Hi, I'm Bryant for your tutoring high school biology. Today's topic, meiosis and chromosomes. Before we start meiosis, I'm going to talk a bit about chromosomes. Chromosomes are really super tightly packed DNA. Let me explain how it's done. You start with a DNA strand, and that gets wrapped around something known as a histone. This mix-up creates a nucleosome. This combined histone and DNA is then super tightly coiled with other of those nucleosomes until you get something that looks kind of like a head with bunny ears coming out of both ends, what we tend to recognize as a chromosome. Humans have 46 of these, 23 pairs if you will. One pair is devoted to sex. One X and a Y makes a boy, an X and an X makes a girl. All the others are known as autosomes. You'll need to know that for when your teacher starts bringing out karyotypes, a diagram of all the chromosomes in the human body. Also, it's good to have an understanding of what a chromosome is when we hit meiosis, which we'll hit right now. Meiosis is a lot like mitosis, but the big difference is, whereas mitosis just makes a copy of the cell, meiosis produces gametes, or sex cells. These sex cells have half the number of chromosomes as a regular cell. That way it can meet up with another sex cell, and they'll have the same number of chromosomes as a regular cell, and produce offspring thereby. Let's get into meiosis. It starts out in prophase 1. Why prophase 1? Because there'll be a prophase 2 later on. Anyway, in prophase 1, you see that the chromosomes have again appeared, but this time they've linked together in tetrads. Let me explain. The chromosomes that link together are homologous chromosomes, if you will. They code for more or less the same trait, such as skin color, eye color, hair color, maybe even personality traits. The linkage, known as a tetrad, allows them to do something known as a crossover, or swap parts of themselves that code for the same stuff. This allows for greater variation, and over time, evolution. Over here, the centromeres are doing more or less the same thing they did in mitosis, preparing spindle fibers, getting ready to head into metaphase. In metaphase 1, the chromosomes again line up in the center of the cell, but this time they're in tetrads. In anaphase 1, they get pulled to opposite ends, and we hit telophase 1, just as we did in mitosis, when cleavage starts to form. Prophase 1, they split apart into two cells. Now, you notice that we skipped interphase. There was no interphase between telophase 1 and prophase 1. That means the DNA was not doubled. Then we'll head into metaphase 2. Now, I'm going to show it to you with only one cell, but just keep in mind that there are two cells doing the same thing simultaneously. In metaphase 2, we have the chromosomes line up in the center of the cell. Keep in mind, this is half the usual amount of genetic material, because we skipped over S phase before heading into the cell cycle again. In anaphase 1, again, the chromosomes are pulled to both ends of the cell, and finally, telophase 2 cleavage forms in those two cells, and we get 4 cells, each of them with only half the number of chromosomes as usual. These will then fuse with a sex cell of the opposite sex. Now, one difference between male and female gametes. In males, all 4 of these will be used. In females, however, 3 of these will donate their cytoplasm to the third one to make a really big cell. This is an egg cell, an ovum, and the reason it needs all that cytoplasm is because it will need to nourish a developing offspring. Anyway, that's females. To recap, chromosomes are super packed DNA. DNA is wrapped around histones forming nucleosomes, which are again supercoiled into forming chromosomes. Humans have 23 pairs of chromosomes, 46 chromosomes. One pair is devoted to sex. X and X makes a girl, X and Y makes a boy. You'll need to know this for karyotypes when your teacher shows them to you. Meiosis produces gametes, sex cells. You go through prophase 1. In this, homologous chromosomes that code for the same information will join and form tetrads. They're swapping parts of themselves in a process known as crossover, so that there's more variation. Then things head along more or less like mitosis, and you reach telophase 1, the cell splits off. However, you skip an interphase and do not double the amount of DNA, instead going straight into prophase 2. Metaphase 2, the chromosomes again line up in the center of the cell, but this time they're only half the regular number of chromosomes. Anaphase 2, they're pulled apart, and finally, after telophase 2, you end up with 4 gamete cells, each with half the regular number of genes. In males, all 4 are used. In females, 3 donate their cytoplasm to the last one to form an ovum. Alright, that's all for now. Again, I'm Bryant. See you next time.